Okay, so essentially, the earlier panels session, they actually asked me about hope. And I think hopefully after my talk, you'll actually understand what I mean by hope, and that this is the best time to be doing brain cancer research because we are making a difference. So let's just get started. So glioma. I'm going to actually talk about low-grade glioma and high-grade glioma in this talk, but we definitely know that glioma affects all ages. It affects both men and women, so unlike breast cancer that only affects women, it affects both gender. There's no preventative screening measures, and as I mentioned earlier this morning, we still don't know what causes a glioma. And there's been no effective treatment strategy other than the STUP protocol, which is the radiation with temozolomide. So I thought I'd actually go through a few of the reasons as to why brain tumours are so difficult to treat. Brain tumours are highly invasive. So as treatment improves, we're starting to see brain tumours popping up in other locations other than the original location. So we need to find a chemotherapy that's not only going to kill the cells, but also target those invading cells. They have a high resistance to therapy, and you would have heard the name MGMT mentioned a few times today. And what the problem is that temozolomide actually attacks the DNA in cells, so it breaks the DNA. And MGMT actually goes along and repairs that damage. So in effect, you have no effect. And so patients with low levels of MGMT actually do better. It's because they're lacking this protein and the temozolomide can work much more effectively. And that's what we refer to as MGMT methylation. It's the lack of that MGMT that's a good prognosis. The other thing that we've got an issue with is once we get the drug in, the tumour has this great facility to spit the drug out. And this is what we call multidrug-resistant proteins that cancer cells have in their membranes that actually pushes the drug out. So we've got to address this issue too. One of the biggest issues, though, by far, is the genetic variability. And as Michael Buckland showed you before with these pictures, these are H&E pictures, to me they all look like glioblastoma. They've all got microvascular proliferation, they've all got necrosis. So the diagnosis is glioblastoma. However, when you look at the genetics, and this is what I term the genetics, where we can see things that are overexpressed, and they're represented in red, and things that are underexpressed, which are represented in green. Now, those three samples that I showed you actually sit in different parts of the genetics. So we had one GBM that was a very poor survivor, whereas one of the other GBMs, which we couldn't see with pathology, was actually a long-term survivor. So we need to integrate pathology techniques with new molecular techniques as well. And this is where our lab comes from, is that to us, it is all about your genome. Now, if we can just stop with the low-grade gliomas for a little bit, and Michael Buckland did touch on this in his session before, but low-grade glioma is very complex, and we do have molecular markers that can help distinguish an oligodendroglioma from an astrocytoma. And it's important that we do this because the prognosis is different, as well as the treatment. Oligodendrogliomas with a loss of 1P and 19Q do extremely well with chemotherapy. So it's important to know this. So the new look, which will be discussed at the next World Health Organization meeting, is to actually use additional molecular markers. Now, the majority of low-grade gliomas have a mutation in IDH1. And as Michael alluded to before, is that we do actually need to go back to Year 12 biology and understand the Krebs cycle again. But with IDH mutants, they produce too much of this chemical called dehydroxyglutarate, which then causes methylation and then causes the tumour. You don't need to worry about that, is that if you have a mutation in the IDH1, which is usually associated with low-grade gliomas, the prognosis is good. What I'm interested in is actually this forms a new target for treatment, which I'll go to in a second. What we're finding, though, with the molecular biology workup is that oligoastrocytomas are probably going to be removed totally. And so we'll either, you come into the clinic, it looks like a low grade, we'll test for IDH1 mutations, and then we'll test for a couple of other markers. And these are mutually exclusive. So for the oligodendroglioma, most of these have loss of 1p19q. They also have a mutation in TERT. Now, TERT is a telomerase enzyme that actually makes the cell survive long term. So they've got a mutation in this, and that actually keeps the cell dividing. So again, it's another target we can get. With the astrocytomas, they tend to have a loss of p53, which is a tumour suppressor gene. And they also have mutations in ATRX. So now, diagnostically, we can use these molecular markers to actually pinpoint what the diagnosis is and be more specific about the prognosis. Now, I mentioned IDH1, and I think this is probably the most exciting thing to actually reveal. So IDH1 mutations were actually found in 2008. So only in that small journal called Nature. But I was in San Diego a couple of weeks ago, and I think this is probably the most exciting news that I've seen in a long time. And that was that on March 24 of this year, the first patient with an IDH1 mutation was put onto a phase 1 trial for the new compound called AG120. Now, AG120 is a targeted treatment directly against the IDH1 mutant. There is also a brother therapy at the moment for the IDH2 mutations, and that's actually moving on to a phase 2 trial. So it's early, but I think it's damn exciting that there's actually a target out there for IDH1. Now, GBM, our good friend GBM, is a little, I wouldn't say it's more complex, but it's a very different disease to a low-grade glioma. What we're finding now is that there's not one form of glioblastoma. There's multiple forms of glioblastoma. And I think this is one thing we need to work with the pathologist to actually subtype these GBM a little bit better than what we're doing at the moment. Now, they don't differ in survival, but we are starting to find, anecdotal at this stage, but we are starting to find that they differ in their treatment outcome. So as you can see here, this is what we call a Kaplan-Meier curve, which is a survival curve. And we've got this group up here which are doing really well. And they're called a proneural subtype of GBM. And they are younger in age, and they tend to be what we call a secondary glioblastoma. So that means that they've come up from having a lower grade glioma and then moved on to a glioblastoma. But these patients do have a very good outlook. Now, if we can actually subtype these patients and then target their treatment better, that good outlook is going to get better. And that's where I hold the hope for. Okay, so what we do in the lab, so just going back to, as Sue said, I'm the head of the Cure Brain Cancer Lab. And what we do is we work hand in hand with the clinic. So when a patient comes, they have surgical removal of their suspected tumour. If it's a GBM, they undergo the Stupp protocol, which is radiation plus temozolomide. Unfortunately, in most cases, if not all cases, the tumour does relapse. Now, what we do in the lab is what we call dynamic biobanking. And you can meet the dynamic people over there, sitting in the back there. But what we do with dynamic biobanking is we go up to surgery and we take the tumour sample, we dissociate the cells, and then we either have them as a cell model or we put them in the mouse. Then what we do is a genomic screen, which is what we're currently doing at the moment, but also a cancer drug screen to try and match that treatment with the patient. Hey, get back here. And then hopefully what we want to be at is to be able to do this process fast enough so that we can report directly back to the clinician and say, okay, patient A sample did really well against treatment X, and therefore I think you should give it a go. That's the long-term aim of our lab, but at the moment, if we can do the preclinical workup for drugs and actually show that a pharmaceutical drug, which may have some efficacy in 10%, we can actually tell the pharma company it's this 10% that are going to benefit. And that's how we're going to improve survival. Okay, MGMT testing, as Michael alluded to, his lab does MGMT testing, and so do we. MGMT is very clinically important, but it's something that's not used at the moment because there's nothing to treat the other arm. So back in 2005, Monica Heggie showed that if you're MGMT methylated, the survival was 22 months compared to those that were unmethylated for the MGMT, which was only 13 months. And I think one of the instrumental things was at 2 years, the survival was close to 50% in these patients with MGMT methylation. So that's why it's such a significant marker. I think even more so now is the use of MGMT in the elderly patients. And you can see from these survival curves again that if you're MGMT unmethylated, so that means you've got too much MGMT, there really is no point giving these patients temozolomide. Just give them the radiation and they'll benefit from that. However, with this group of patients that are MGMT methylated, which means they have less MGMT, if you actually give them the radiotherapy plus concurrent temozolomide, there is a significant improvement in survival, so it's worth pushing the patients through that. Okay. So one of my little bugbears at the moment is the MGMT unmethylated patients. We've got to come up with something better than temozolomide, surely. You know, if we all know that there's minimal benefit, why do we give it? And I think the short answer to that one is that at the moment there is no alternative. So I've directed our lab to work in this space of MGMT unmethylated patients. And again, here's Eric, you may have recognised from this morning, and you can bombard the lab over there again. What we're doing is the preclinical workup of several drugs that have come either from commercial or pharmaceutical companies, but we also have a drug that was made in-house at UNSW as well. And we're using these patient models and using radiotherapy to try and work out what drugs are going to work on our patients in the MGMT unmethylated setting. Now, one of those drugs is called veliparib, which is a PARP inhibitor. Don't get too worried about the science here, but essentially it's a little bit like temozolomide and the MGMT story, but it's a different arm of the DNA repair. So if you give a PARP actually repairs the DNA of single strand breaks, and the cell survives. So what we want to do is get rid of this PARP, and so we use a PARP inhibitor. Okay, and so what we did was we screened a whole heap of cell lines, and then what we did was we found the ones that were most resistant to the drug and the ones that were most sensitive to the drugs. And then what we did was a genetic screen to work out, you know, what was different between the resistant and the sensitive cell lines. And what we found was a PARP inhibitor sensitivity signature. What's interesting about this signature is that it's actually the same signature as what they use in lung cancer as well. So there's some similarities. In breast cancer, BRCA1 is a sensitivity gene for PARP inhibitors, but we do have a sensitivity signature now in GBM. And I've been coupling the PARP inhibitor with radiation, and I've just circled the two cell lines that actually have that sensitivity signature. And as you can see just by looking at the yellow bars, which is radiation only, versus the red bars, which is the combination treatment, the combo is doing better in these sensitive cell lines. So, a cell's a cell, but a patient trial is exciting. And we've just had the green light from AbbVie to actually move forward with the phase 2 trial. And what we'll be doing is, this will be upfront in glioblastoma. They'll be tested for MGMT. The methylated patients will receive the standard treatment because that's where they'll receive benefit. But for the unmethylated MGMT patients, they're going to receive this new drug, which is veliparib, concurrent with the radiotherapy. Now, what we're going to do in the lab is actually take the patient tissue and test it for the sensitivity signature. We're going to subtype the GBM to see if there's certain subtypes of GBM that are more responsive to the PARP inhibitor. And one of the things that I think is really exciting is looking at exceptional responders, and I'll come back to that point in a sec. So another drug that we've actually designed in-house, and this was by Professor Phil Hogg at UNSW, is called PNAO. And again, going back to that boring Year 12 biology class, brain tumours do actually have a lot of glycolysis, so much more than what normal cells do. And one of the drivers of this intense glycolysis is this gene up here, which is hexokinase 2. Now, what hexokinase 2 does is it binds to a channel, and this is how the tumour cell actually makes energy. So the obvious thing to do is stop the cell from making so much energy. Shut it down. And this is what we've made, which is a drug called PNAO. It's an arsenical compound. And this is just some early animal studies to show that it works as a xenograft model. But where we're really having fun is actually putting PNAO together with radiotherapy, and PNAO in combination with a compound called DCA, which you may have heard from Canada. So in combination, we're finding some really interesting efficacy. So watch that space. And again, it's in a clinical trial setting at the moment. So essentially, and I think this is where we're at now, there's a lot to be learnt from history. And I think one of the things that we do need to take from history is the fact that, okay, clinical trials have come and they've gone and they've failed. But I don't think that's the case. And what we need to do is look back over those clinical trials, because I bet in every single clinical trial, there's always that one exceptional patient or 10% of exceptional patients. What we need to do is go back and look at those clinical trials and say, okay, here's the exceptional patients. Why did they perform well? And I think this is key to it. Why did they perform well? And this is what we're doing with a lot of our studies now, is looking at the exceptional responders. And Kerrie here is looking at the Cabaret study, which was looking at Avastin plus carboplatin. And we do know that it didn't work for everyone, but it did work for some people. And so we're trying to unravel the genetics in those patients. Can I ramble on just 1 minute? Okay, I couldn't not put cancer immunotherapy up, and I think Brenda said it this morning too, that she's getting a lot of questions about dendritic cell therapy. It's a tough one, and I think the bottom line is it's too early. It's done wonders in melanoma, there's no doubt about that. But for brain cancer, where we know it's not caused by an infection, we know it's not caused by a predisposing factor, it's a little bit too early to show that cancer immunotherapy is working. So this is the dendritic cell vaccine that has been long around, and that's essentially taking the tumour, mushing it up, taking some mRNA as well, and then you drive it into the dendritic cells and then pretty much put it back in and you're vaccinated. I've done a review before this talk today. I did a review on all the immunotherapies, and to be honest, the results aren't there. I mean, there's a lot of hoo-ha and a lot of greatness going on with the companies, but the evidence is not there. The other thing that we've been looking at is chimeric activated receptors, or CARs, and this is genetic engineering. Now, there may be evidence for this one here, and essentially what you do is you genetically engineer a part of that cell, and essentially it then targets the tumour cell where you have a receptor complex, and then it will kill the cell. Now, the CARs are doing wonders in leukemic cells, and there is some evidence to show that it may be working in brain tumours as well. I guess watch the space, never say never, and let's hope it works. Collaborations, we're only as good as the next person, and I'd have to say in Australia, collaborations is at the forefront, and so are acronyms. And yeah, as I said, there's my lab over there, so if you've got any questions, by all means ask me or them, put them on the spot. Okay, thank you.